Now, for the plasma membrane, we understand that it surrounds the cell and protect it from the external environment. So it's separating the cell from the external environment. But also we understand that within this external environment, there are things that the cells need, things that like food, cells need to bring that food inside them. And also there are the wastes, cells need to send them outside the cell. So this means that this membrane is selectively permeable. What does that mean? It allows certain materials to pass through it, whereas it prevents other materials and keeps them outside. So selective permeable or selective permeability, it's that its ability to allow certain things to pass and to prevent other things from passing. And these things depends on the cell's need, whether they need these materials or not. Now the plasma membrane is, if you remember, it's a lipid bilayer membrane and it's made of the phospholipids, which are made of the polar head and non-polar tail. And the way it is arranged, the tails are faced together, whereas the heads are away from each other. So this is a phospholipid, and these would be like this. If we go back, as you can see, this is the head, this is the head, these are the tails. Okay, so this is, what does it mean? Biolipid. It has two layers of biolipids. Also, it has cholesterol. Cholesterol, it makes the membrane a little bit rigid, a little bit hard. Also, it contains proteins that these would be means of transport. They will form channels. And carbohydrates, these are to recognize, like for communication. So if we go back into this is the cholesterol, these are the proteins, and these are the carbohydrates. So all of these four things are part of the plasma membrane. Now there is this video, please watch it and see. It will help you to understand how the overall structure of the plasma membrane. Oops, I want to go back. Okay. Next, now we need to understand also a few concepts about how the molecules are crossing the plasma membrane. And for these molecules to pass through the membrane, we have two options. Either it's a passive transport, and in this case the cells do not use energy, so they don't need to expend energy, and that is passive transport, or it is active transport that cells must use energy or spend energy for that transportation. These are the two major types. Then there is a subtype, it's called bulk transport. Technically it's part of the active transport, and here it involves these vesicles, the trucks we talked about, and these to move large substances. We have exocytosis and endocytosis, and we will talk about them. But let's start with the passive transport. With the passive transport, we have two major types. One is diffusion, and the second one is osmosis. So what is diffusion? Diffusion is the movement of these molecules from regions of high concentration to a region of low concentration. Think of, let's say, for example, let's use the notepad here. Okay. So, for diffusion, I need to pen. So for diffusion, we said this is the movement of solutes from areas of high concentration to low concentration, right? What does this mean? Think, for example, if we have this cup of water and we dropped within it a cube of sugar. Okay, after like a few minutes, sugar would start to diffuse, and additional more minutes we will have the sugar all over the cup. That is diffusion. The sugar by itself moved from the areas of high concentration to areas of low concentration. This is high concentration because this is the cube of sugar is here. This is the area of low concentration because there is no sugar here. So the sugar moved from this area toward these areas, the areas of low concentration, till the moment that the sugar is filling all of the cup, all of the water in the cup, and that is diffusion. And as you could see and understand, there was no ATP used for this sugar to transfer or to move around. It's not this one, not this one, here. So that is diffusion. So for the plasma membrane, substances, sometimes this is outside, this is inside. Sometimes we have substances here outside, they are in high concentration, whereas here inside they are with low concentration. So they will move from areas of high concentration to areas of low concentration. That is diffusion. And this movement from high to low is the movement down their gradient. Down their gradient. They moved from the area of high to area of low concentration, and that is diffusion. And as you can see here, for example, this dye at 10 minutes, it's like this, 1 hour, it's like this, 24 hours, it's like this. So there is a kind of diffusion in water of this dye. Now, sometimes this diffusion can happen through the plasma membrane. Sometimes it needs a channel, like a protein, to move it. And that is diffusion through channel. Or sometimes it's called facilitated transport, that it needs a transporter to move it. Think of this like elevator. It picks it from, or a crane, it picks it from one side and release it into the other side. Still, this is diffusion. These are moving from areas of high to low. The difference is there is something that is transporting them. Okay? Here, think of it as escalator. So it's always open, and it helps in moving, again, the same concept, from areas of high to areas of low concentration. Here, there is nothing need to help them. These particles, they are so small, they don't have any problem with the plasma membrane. They can diffuse through the membrane. Usually these are lipid soluble. Okay, whereas these are not lipid soluble. These are some polar molecules. For facilitated transport, these are large molecules. We can't simply pass them through here because they would disturb the plasma membrane. So these are the three types of diffusion: direct diffusion, or just diffusion through the lipid membrane, diffusion through channels, and facilitated transport. Now, the other form of diffusion is the diffusion of water, which is osmosis. Here also it's happening across selective permeable membrane, and of course even here this is selective permeable. It allows certain things to pass and prevent other things. So still, selective permeable is always, the plasma membrane is always selective permeable membrane. But the water would move from area of low solute concentration to an area of high solute concentration. What does this mean? See, for example, here, these two chambers they are connected through this membrane. This membrane, the pores here, they are so small to allow these sugars to pass through. They cannot pass through. So the water, and in this case, this is the area of low concentration, of course, and this is the area of high concentration. If the membrane, its pores were big enough, then the sugar would move in this direction. But that is not happening because the membrane, its pores are so small. So in this case, water would move from the area of low concentration of solutes to the area of high concentration of solutes. And we will end up with difference in water volume, but equal concentration of sugar. We'll end up in difference of water volume, but equal concentration. A direct application that I think usually or daily doing it is the tea bag. So this is the tea bag. The tea bag, it has these little pores, and the tea leaves are inside. So when you put it in a cup, you put the tea bag in the cup, the leaves cannot leave the cup, right? But the water will get inside and like kind of solve or dissolve the tea, then the tea would come out. But that movement of water from area of low concentration to the area of high concentration within the tea bag, that is osmosis, and that is a direct application, something that we usually do when we are preparing our tea. Now, because of the selective permeability of the membrane, we have, what is it? We have different in something called tonicity. Some different in tonicity. And I will go back to the OneNote to explain this. Okay. With different tonicity, what do we have? We have the cell. Okay, this is the cell. And the cell, of course, is sitting within an environment. Okay? Now, what usually happens that within this environment, there are materials outside and there are materials inside. In case they are equal inside and outside, we will call this isotonic, in which concentrations are equal inside and outside. But if these materials are concentrated outside, only few inside, we will call this hypertonic. Hypertonic, high concentration outside. What will happen in this case? That the water here, this is the area of low concentration, would move to the area of high concentration. Right? Water would move from the area of low concentration, which is inside the cell, to the area of high concentration, which is outside the cell. So the cell will lose water. And will shrink. Right? Now, let's see what happens if it is the other way around. If there are high concentrations inside the cell and this concentration outside the cell. What will happen in this case? Now, the water outside will get into inside because the water again is moving from areas of low concentrations to area of high concentration. So in this case, this we will call it hypotonic. Hypotonic. With hypotonic, concentration inside is higher than outside. Is higher than outside, and water will move in, which will lead to cells swell. Okay, so these are the three different types of tonicity: either isotonic, or hypertonic, or hypotonic. And here, something we did not talk about, there is no net movement of water. Water would move out and in without causing anything to the cell. But with these, with the other types, like here and here, there is a net movement. Either the water would move out or the water would move in, and this will either cause the cells to shrink or to swell. And these, the hypertonics, the hypotonic, and the isotonic, of course. Now, why these are important? Because when we are transporting blood, for example, it's extremely that we put the blood cells in isotonic solutions, and that's the normal saline. Otherwise, the cells the red blood cells either going to shrink or going to swell. This video shows a home experiment about osmosis, and it will help you to understand it better. It's very funny, and I advise you to watch it. Okay, I will stop here, and when I come back in the new part or in the coming part, I will talk about the active transport.